Okay, thank you. It's a great pleasure to be here. So I'm going to talk, so the last talk we saw beautiful imaging looking at the dynamics of proteins. And so I'm going to talk about how we're going to try to feel the dynamics of the proteins using forces. And it'll be two different stories, one about polymerases, E. coli replicative DNA polymerase, and another about HIV replication, both of which are really related to things going on tracks and encountering obstacles. And so let me just start with the basic. So I'm going to talk about measurements, like I said, feeling the protein interaction. So we use single DNA molecules that are captured between two polystyrene beads. And in the talk here, I'm going to talk about very long DNA molecules and look at sequence averaged DNA binding or polymerization effects. And so we have in this study, we have a glass micropipette, which allows us to move the bead and stretch the single DNA molecule. And that bright spot is the optical trap that's holding the other bead in place. And as the DNA is stretched by moving this micropipette, this bead will move slightly relative to the position of the trap. And that distance that it moves is proportional to the force. And so you can detect things like protein binding events or replication of DNA by detecting changes in length or forces on the DNA due to those interactions of the DNA. And so this is a schematic diagram showing the DNA molecule, and here's what you see in your image. When you look into one of these microscope objectives, and here I'm plotting the force in piconewtons versus the extension in nanometers per base pair. So what we do is we take our long DNA molecule and we divide by the number of base pairs so that this graph that we get will not depend on the length of the DNA. So if you stretch a DNA that's 50,000 base pairs, you get the same curve as if it's 20,000. If you go down to like 100 base pairs, then you start to get big differences. But as long as it's a long polymeric DNA, you get the same result. And so as you stretch the DNA, when you reach about 0.34 nanometers per base pair, that's the distance between the DNA bases. And so what you've done is you've stretched out the entropy essentially out of the polymeric DNA molecule. And then you begin to see the force go up, and then you're stretching on the DNA double helix itself. And so that measures the elasticity of the double helix. And so what you're doing is applying a force on the DNA, this double-stranded DNA, and eventually you get to a point where you get this overstretching transition, and there's going to be a whole talk this afternoon about overstretching, so I won't go into the details. Here I'm showing it as basically denaturing the DNA and melting the base pairs, and that depends on solution conditions exactly what you see. There's a whole phase diagram that people haven't really developed yet, but we know that sometimes it melts the DNA and sometimes there are other structures that form, but those structures are very close in energy to DNA melting. And so if you keep going, you eventually create mostly single-stranded DNA. And so the important thing for this talk to know is that as you increase the force on double-stranded DNA, you're destabilizing the DNA. And that destabilization will help processes that convert double-stranded DNA into single-stranded DNA. And one of those processes is polymerization. And so I'm going to start out talking about a story about the E. coli replicative polymerase Pol III core. And we can use force to switch it between exonuclease and polymerization activity. And so we'll talk about how we can control that activity. And then I'll talk about a DNA binding protein involved in HIV replication that actually acts as a roadblock to polymerization. So to start with, in E. coli, there are actually at least 5 polymerases. And this really complicated one is Pol III. So this is the holoenzyme complex. You see it has a lot of different proteins involved. And there are also other proteins that are important for mostly dealing with lesions in the DNA, with DNA damage. And so this will have to get switched out with other proteins, these Y family polymerases, for example, in order to deal with DNA damage. So you have a polymerase switching process, which is something we're interested in studying in the future. But right now I'm going to focus on this Pol III. Okay, so why is it so complicated? Because during replication you have 2 strands, and you can move, the polymerase can only move along the strands in one direction to replicate the DNA. And so that's called on the leading strand, that is what happens. And on the lagging strand, you have to form this big loop. and you have Okazaki fragments, and you go through in the reverse direction, and you do one fragment at a time and loop around. And I'm not going to go into the details of describing it because it's not important. We're not going to study that looping process. But this is just to give you a bigger picture about what happens in E. coli replication. So among all these motor complexes, here's Pol III core, and that itself is also 3 proteins. And so we're going to zoom in and start just looking at this Pol III molecule by itself to study it. And so it turns out that this has these 3 components, and one of them is the polymerase called alpha. And that's the thing that does the polymerization and adds the bases to the end of the strand that are complementary to the other strand. And then epsilon is the proofreading enzyme, and so that does the exonucleolysis. So that has it go backwards when it encounters the wrong base. So occasionally it will make an error and put the wrong base in. That's not complementary, and the rate of making that error is already to the -5. So it's pretty rare. So it's much more accurate by itself than the ribosome is, for example, during translation that we just learned about. Now, with proofreading, with this exonuclease activity, is actually 10 to the -8 per base pair. So it's extremely accurate, and that's very important for faithful replication of the genome in E. coli. So our experiment that we're going to do is try to understand how this exo and Pol activity it can be regulated. And so it was discovered almost about 15 years ago that you could actually take a polymerase, and this was done with T7 in the Bustamante lab. They found that if you take a polymerase and if you have a construct, say, optical tweezers instrument like this, where you have a free 3' end, which normally a polymerase will come and start polymerizing from that end and add complementary bases. If you have that construct, you can see the polymerization by simply adding nucleotides and the polymerase. And if you hold your tweezers at constant force, you can measure the change in length as it polymerizes. So you start with something that's partially single-stranded DNA, or you could imagine starting at almost fully single-stranded DNA. And as it polymerizes, the length will decrease until it reaches the double-stranded DNA, fully double-stranded DNA. But what they found that was more surprising was that if they went to a high enough force, they could get it to undergo exonucleolysis, where it goes backwards and chews away at the strand of DNA and forms single-stranded DNA. And so you can control that activity by going up and down in force, and they could reverse the process. And this was a T7 DNA polymerase, which is a single protein that can switch between those 2 domains that have those 2 different activities. And so that's kind of similar to our experiment, except we have these 3 proteins. There's 3 protein complex, and they're actually all separate proteins. And one of them is alpha, one of them is epsilon, and one of them is called theta. And it doesn't really do much except hold the other 2 together that we know of. And so, these are experiments that we did. And so we found that, for example, if you add this, so this is Pol III core with these 3 proteins, and you see that if we go to a high enough force, we can get it to undergo exonucleolysis, and then we can switch it and go to a different force, and it will still undergo exonucleolysis, et cetera. Then we go down and we get to some force where it's not really going to do much. If we keep decreasing the force, then we start to see polymerization, and then more polymerization until you get to almost fully double-stranded DNA. And so if you look carefully, so what we're doing is we're stepping through the forces, and we're observing this exoactivity. And the other thing that you notice, besides the fact that it's either going one direction or the other, is that the slope of this is changing, right? And so that slope change represents a change in the rate of exo, or the rate of polymerization activity with force. So force regulates its ability to polymerize and undergo exo. It's not just switching between the 2 regimes, but it's actually changing the rate at which it does that. And so if we zoom in on one of those, so one of those exo at a particular force, we see, here's our data with some noise, and you can very clearly see steps. And those steps represent either exonucleolysis or pausing, or exo or pausing. And you can do a sort of a window averaging. And within that average, you can look at the velocity, and you get clearly an average. There's sometimes when it's 0, and there are other times when it's on the order of tens of nucleotides per second on exonucleolysis. And so that's one thing that you can study is the force dependence. But we found more interesting was the fact that there were these steps. So these are pauses in the motor activity along the DNA molecule. So what causes those pauses? Is it pausing of the motor? Is the motor dissociating and waiting for another motor to come on? So that was the question we were trying to answer. And so if you want to know if it's waiting for another motor or not, or just pausing by itself, you look at the concentration dependence. And so if it is dependent on concentration, that means it's waiting for another motor to come. And so we saw, indeed, at low concentrations, the pause time was high, and then as we went to higher concentrations, the pause time went down. And so that means that the pausing was induced by waiting for another polymerase to bind. So unlike moving along microtubules, this protein is doing its activity, dissociating. Another protein is coming and doing a burst of activity, dissociating, and another one bursting and dissociating again. The dwell time, the time that it stays on when it's doing the exonucleolysis, does not depend on concentration, as you would expect. So it does its activity for a certain amount of time and dissociates spontaneously. So how can we describe this quantitatively? What's really happening? So there's some initiation rate for an exo event after a pause. And so that rate depends on concentration, and so that's really just 1 over the pause time. Okay, so that's the rate of initiating these events. And so we can fit this to a model. So this is a mechanochemical model. Basically what we're saying is that, okay, there is some bimolecular step, so there's some probability of binding and undergoing this exonuclease, of binding to the DNA to the place where it can undergo the activity. And that is, of course, proportional to concentration. The more you have in solution, the more frequently it binds if the site is open. And then there's some K minus 1. And then after it binds bimolecularly, it has to go into a state that is competent for exonuclease activity. So there's some non-concentration dependent conformational change, and that rate is described by K2. And if you assume that this rate is in pre-equilibrium to this state, then you can write this equation like this, where you have essentially the population of this state feeds the population of this state. And so you can write that so that K2 depends on K1 by this cofactor here. And you can write that in terms of the binding affinity. And if you then fit it to your data, you can get a rate of going into that state if it's already bound to the DNA. And you can get a KD for binding to this state. And that KD for binding can be compared with biochemical experiments, and we get the same value. Yes? Does the force have any role in the pausing? Well, if the pausing is due to the so the pausing is due to dissociation. So
does the force affect this rate?
Right. Yes. So we'll go into that in a second. So that's actually what's coming up here. Sorry. So here's our pause and dwell time, and versus force. Okay. So yeah. Sorry, I was mixed up. So this is all the concentration dependence, and you can fit the concentration dependence, and this is all at 1 force, which I think is 30 piconewton. So next, what we're going to do is go into the force dependence of these rates, and the pause is strongly affected by force, but the dwell is not. So again, so once it's on there, it's going to move at a certain amount, and that does not depend on force, just like it doesn't depend on concentration. So how do you expect this to depend on force? So here we're plotting on a semi-log scale, and so here's the force, so you expect some kind of an exponential dependence. And so this go back to the beginning where I was talking about how applying a force destabilizes the DNA. So you're doing some amount of mechanical work if you can convert the length from double-stranded to single-stranded DNA, or the reverse. And that amount of mechanical work is essentially the area under the curve at a given force. And so the amount of energy required to break destabilize a single base pair is this delta G, is this integral under that curve. And delta G0, we're assuming here, for the purposes of this analysis, that at the overstretching transition at 62 piconewtons, that it's basically single-stranded DNA, okay? And so if that's the case, you can calculate the initialization rate is Kf times e to the n delta G over Kt, where delta G here is the difference between the delta G of force and the single-stranded free energy to make single-stranded DNA. So it's how much you're destabilizing the DNA, basically, is what this delta G is. And so n is the number of bases. So we're saying this is the delta G per base pair, and then n is the number of bases that you're destabilizing. And so if you fit the data to this, you get n is about 2 base pairs. Okay, so what it says is that it seems like if you destabilize 2 base pairs, it starts to go backwards. Okay? And so, so that's how it's controlled. So force is telling it to switch between these 2 different proteins. So remember, before it's going into exoactivity, alpha is there, and then epsilon has to bind and then bind into a configuration that tells it to go backwards. And so, of course, you're not applying these kind of forces in the cell. Those forces aren't being exerted. But this is destabilizing, just like you think maybe mismatches are destabilizing the DNA. And so again, this is our model. And so the idea is that 2 base pairs opening are detected by epsilon such that it says, oh, there are 2 base pairs open. There must be damage there. I'll have to go and cut those bases out. And so to test that, so in the lab of our collaborator who provided us with the proteins, Penny Boyning's lab, also at Northeastern, we said, well, how about if you test and see in your experiments, in your bulk experiments, how does it detect 2 base pairs or 1, or does it, and it turns out that's true, that as we predicted, it really does not go backwards if it only sees 1 base pair mismatch. But if it sees 2 mismatches, it starts to have exoactivity. So this is the exoactivity. And 3 doesn't really increase the exoactivity that much. So that's kind of what we predicted would happen, is that it seems to detect the mismatches by detecting 2 base pairs fluctuating open. Not that they're mismatches, but that they're just 2 base pairs that are fluctuating open, that they're single-stranded bases. Yeah, that's a good point. We didn't really test 1 followed by a match. Either it detects that, and normally that is what happens, or it would be more likely to make a second mistake if it makes the first mistake. I don't know which ones of those. We didn't really test that, but that's a good point, that normally you would think it would have a mismatch. You know, why isn't it advantageous for it to just either detect the first one, or if there's a second one that's matched, still see that it's there? I don't think it would be the same if there's a second one. It depends on how much that means the DNA is destabilized. If there's a second mismatch, it probably frays open pretty consistently. So there's a free energy difference that matters. It might very well matter exactly what base that second one is. It's the stability. It's really detecting the stability of those last 2. So you could add up what that stability is for a given configuration and figure it out. So it doesn't really. The point is, it doesn't depend on what the base itself is. It just depends on how stable those last 2 bases are. Okay, so yeah, so that's basically the idea, is that we can look at exo and pol activity, and we can see switching between these 2 separate proteins, which actually wasn't observed. All the other polymerases that have been studied have been highly processive polymerases that have both exo and pol activity as part of the single polymerase. The substrate stability, it determines, so it's substrate-directed motor activity, right? Okay, so the next story that I wanted to talk about is really switching the biological systems and switching from, I'm not going to study the motor itself, but binding proteins, but we learn about the binding proteins and how they can regulate the motor activity. And so this is about human innate immune system proteins called apobec proteins. So in order to understand this, we have to go back to the biological system. And so this is about HIV replication. And so HIV is a retrovirus. And the retroviruses are viruses that contain RNA as the genome. single-stranded RNA as the genome, and actually 2 copies of single-stranded RNA for some reason. And they do base pair with each other to some extent, but they're not complementary to each other. So you have these 2 single-stranded RNA bases. They enter the cell. There's a protein shell around them. And then each of these single-stranded RNA molecules is converted into double-stranded DNA by a process called reverse transcription. And then that double-stranded DNA enters, so that, again, the virus tricks the cell into allowing the double-stranded DNA to go into the nucleus. There are proteins that are part of the virus that incorporate the double-stranded DNA into the genome of the cell, and then the cell does not know any better but to start making more of the RNA, that's the genome, and also the proteins that are encoded by this RNA genome. And incidentally, part of this process is a ribosomal frameshift. There's a ribosomal frameshift that's designed into the HIV replication that creates the GAG-Pol, the polymerase that it doesn't need a lot of 5% of the time. So that's programmed in to this process. But there are a lot of details about assembly that aren't important for what I'm going to talk about, important for HIV replication. I'm going to now zoom in on the reverse transcription process. But before I do that, let me tell you about this human immune system proteins that give us immunity to HIV. So it turns out there are these proteins called ApoBec3 proteins, and there are actually 7 of them. And ApoBec3G is the one that's best at inhibiting HIV-1 replication. And so, in fact, by itself inhibits HIV replication. However, HIV has evolved to develop its own protein called VIF that binds to ApoBec proteins and tags them for degradation. And so the cell it tricks the cell basically into destroying its own immune proteins. And so it's not useful for inhibiting HIV anymore, but we want to understand how it works. Either you could manipulate it to be more effective, or you could find ways to target VIF, for example. So basically, we want to understand how it works. And it's a deoxycytidine deaminase, meaning that actually it changes the C base to U chemically. And so on DNA, okay, so I told you that it was an RNA genome, but it converts it to the RNA during a reverse transcription. You have a point where you have single-stranded DNA, and so it takes single-stranded DNA and it zooms along single-stranded DNA, and wherever it finds 3 C's in a row, it changes the 3rd C to U, just chemically. And so U is not a normal base in DNA, and so that can target. There are enzymes that come and cut where that is, which can inhibit HIV replication. And also, if you have a U there, you can have mutations, obviously, because you put the wrong base. If this does get replicated, it puts the wrong base on the other strand, and so you get a lot of mutation of the virus because of this ApoBec3G. But only if it's not targeted for degradation. But what's interesting, and what we've been trying to figure out why this happens, is if you take and mutate the active site so that it can no longer perform this process, you have these deaminase deficient mutants, and they still inhibit HIV replication. So there must be some other process. And so this has been studied in human T cells and even at endogenous levels. There have been many questions about whether this is really biologically relevant, but several papers have shown that it is. And in addition to these properties of being an enzyme, it is a really hard protein to work with because it likes to form dimers, tetramers, higher order oligomers, and so if you just take this protein and add it to a solution full of DNA, it drops out of solution. So you can really only do biochemical experiments on very short DNA molecules. And it's actually hard to get rid of the oligomers. Okay, so reverse transcription. I said I was going to zoom in on this reverse transcription process. So the way reverse transcription works is you have this nice straight line RNA. That's a joke. It's not really straight line, obviously. It's a 9,000 base long single-stranded RNA, so it's obviously a very complex structure. But we draw it as a straight line so you can see what's happening. And so it actually takes tRNA packaged from the previous cell and anneals it to what's called the primer binding site, and reverse transcriptase, which can copy RNA into DNA, starts moving along and copying this single-stranded RNA into complementary DNA, which is shown in green here. But strangely enough, it actually does this starting at the very end of the genome. And once it gets to the end here, it has nothing it can do, but it has to go all the way to this other end. And again, because it's actually a flexible RNA molecule, this other end could be very close. But it has to transfer this strand to the other end and then continue its polymerization process and copy it until it creates single-stranded DNA. And there are some more details about how it gets another primer and eventually ends up with double-stranded DNA. Not that important for this talk, but also all a very interesting process. And it makes you wonder how this actually works because it involves so many different steps. And so in the step where you create single-stranded DNA, that's where the deamination of the DNA can occur. And that's at least active for a few minutes that is available. And then our hypothesis is that the deaminase-independent method is one in which it actually will block the polymerase. So it's a binding protein, and we know it can form oligomers. So maybe it will block the polymerization by sitting on the single-stranded DNA and preventing reverse transcriptase from moving. And so that was our main hypothesis for these two mechanisms. And so one problem with that idea is that the A3G, in order to block this, it has to be very slow. And so for this to be a very slow process, that means it has to sit there to block the RT. If it's coming on and off, RT can just wait for it to come off and then start moving forward. However, it can't be slow because it has to scan the entire genome and find all the CCC sites along the genome and modify them for its deamination. And so that requires fast binding kinetics. So it's a contradiction. And so we proposed this roadblock model back in 2007 until 2014, where we actually tested the model in experiments. And the basic idea is that it must start off as a fast binding protein, and then over time form these oligomers. And once it forms an oligomer, it's a slow protein. So this makes sense as a model. And we published a paper showing that it does this a few years ago. And there are a couple things. So one thing is that we said in the paper monomers or dimers, because we didn't know whether they were monomers, whether the fast state was monomers or dimers, and there was a big controversy in the field. Is it monomeric in solution or dimeric in solution? And because it's not a well-behaved protein, it's hard to test these things. So we drew it as dimers, but we said monomers or dimers, but that turned out to be wrong. And we did show these oligomers as these large oligomers, and what we showed is that we measured the kinetics, and we showed that it starts off in a fast binding state with some bimolecular reaction rate, and then it goes into a slow binding state with some other rate, but we didn't know how big those oligomers were. And so, we're doing a single molecule experiment, so we have one DNA molecule in solution, and you've got, Avogadro's number of molecules in solution, and so the oligomers can be as big as they want, right, in our experiment. But in the virus, it only packages about 10 of these. So the problem with this is that this could require 1,000 proteins to make this slow oligomer. And there are only 10 in the virus. So people said, well, this is not biologically, you haven't shown this is biologically relevant process. So we went back and said, okay, we're going to ask these questions. To what extent are these previously observed slow binding kinetics due to oligomerization, and what degree of oligomerization is required, and then how does the oligomerization alter this enzyme activity? And so to understand the oligomerization, we looked at mutants that lack oligomerization capability. So we contacted our collaborator, Linda Celico, who is an expert in studying all the different ApoBec proteins. And she sent us some mutants. So what kind of mutants do we want? Well, here's the physicist's picture of a protein, two boxes, a line between them. So there's an N-terminal domain and a C-terminal domain. And these two domains both are DNA binding domains. Okay, so this protein already has two DNA binding domains that are completely separate from each other. The N-terminal domain is stronger DNA binding domain, and the C-terminal domain is catalytically active. So these are called CD domains, CTD and deaminase domains, because structurally this should also be a deaminase, but it just doesn't function as a deaminase as part of this particular protein. But they're both basically the same kinds of domains. So you have these two domains, and the N-terminal domain can oligomerize with the other N-terminal domain, and the C-terminal domain can oligomerize with the C-terminal domain. So you can see how you can form big oligomers, because you can just keep building on the train of these proteins. Just to put it in context, so there are these 7 ApoBec proteins, and some of them have 2 domains, and some of them have 1 domain. And they all have one deaminase domain, one active domain, and so that's shown in red here. And they also have these VIF binding sites, which is where the VIF binds and targets them for degradation. And some of them don't have VIF binding sites and aren't targeted for degradation, but they don't seem to inhibit HIV replication. So what is the point of them? Well, it turns out the ones that don't inhibit HIV replication inhibit retrotransposon replication. So retrotransposons are these sequences in our DNA that are constantly replicating. And it turns out there is a battle ongoing in our cells to prevent the replication of these long RNA molecules, these retrotransposons. And part of the battle are these ApoBec. A, for example, is a very strong inhibitor of the line 1 retrotransposon. So that gives you a context of sort of the importance of this problem. And some of these other proteins also oligomerize, and we're studying them right now. ApoBec 3H, while it only has 1 domain, seems to also form oligomers. A3A does not form oligomers, is just a very good deaminase enzyme. Okay, so so this dimerization, so we got some mutants, and the mutants are an N-terminal domain mutant that which you call FW, because we mutated these 2 aromatic residues, and this prevents N-terminal dimerization. And the C-terminal domain, we have 2 mutants that prevent the sort of weaker dimerization of the C-terminal domain. And so we're going to look at both of these kinds of mutants and see what happens. And so first of all, are these really mutants of the oligomerization? And so we used AFM to make images under certain conditions of wild type, FW being the N-terminal domain mutant and the 2 C-terminal domain mutants. And you can see differences in the images. Clearly they're different. We can actually quantify them by looking at the size of these bright spots. And it turns out for the FW, there's a big peak right here at the monomer size. And for the this 2 C-terminal domain mutants, there's a big peak at the dimer size. And then the wild type, there's no peak at all. It's just a bunch of things. And the average size is on the order of 15 to 20. Okay, so wild type forms big oligomers. That's clearly the case under the conditions that we've done this experiment. So clearly the mutations have worked, and we have the ability to have something that only forms monomers, something that forms mostly dimers, and something that forms big oligomers. Okay, so the experiment that we're doing is stretching our DNA, and this enzyme works on single-stranded DNA. So what we actually did was take a double-stranded DNA and stretch it up to forces where it becomes single-stranded. This is experiments that we did about a year ago, and now newer experiments, we're just taking single-stranded DNA to begin with to do experiments. But historically, because we were looking at using the annealing to look at the oligomerization, we used this method. But you don't need to have the double-stranded DNA there. So what we did was we went up to these forces where we have basically single-stranded DNA, 80 piconewtons, And then we have our single DNA molecule that we've captured in between the beads. We flow all the other DNA molecules out, and then we flow in a solution containing this A3G protein at a fixed concentration. And then we see that the length changes as the DNA becomes shorter as the protein binds. So this protein binds single-stranded DNA, and so it contracts the single-stranded DNA as it binds. And so we see it do that for some amount of time, and then we can stop, and then we can flow all the protein out of the solution surrounding the DNA molecule, and then we should see it just dissociate, and when it dissociates, there's no protein in solution, so it doesn't come back on, so eventually it should all come off. It doesn't all come off, and that says that on the time scale that we do the experiment, we have some protein that stays bound, and that we think is forming oligomers. Okay, so this is our procedure we can look at association process and dissociation, and to try to figure out what is happening with the protein. So we were kind of surprised in this experiment initially that we could actually see oligomerization in real time in our data. So here is the extension versus time. So if you go back to here, so this is the extension. So as this moves to the left, as a function of time, that's the decrease in extension versus time. And so we see the decrease in extension versus time at a fast rate and then a slow rate. And so you can only fit this to a 2 exponential process. If we stop at some point and then flow in the clean buffer where there's no protein to bind, we see dissociation in what looks like a single exponential process. So what's happening is you start off with your template of single-stranded DNA. You have A3G binding. After it binds, it starts to form these oligomers. And then when we let it dissociate, not all of it dissociates because some oligomers stay bound. That's our hypothesis. But you have to do a quantitative analysis to show this. So here's the FW. Okay, so that was wild type. Here, all this is just a wild type protein, the one that forms the big oligomers. And this is the FW, forms a single exponential. And the fraction bound, it seems to be able to shorten the DNA a lot more if it's only can form monomers. And so you can fit this wild type to the 2 exponential process. And I'm going to call the 2 rates that we get K fast and K slow. Okay? And so there's a fast rate and a slow rate. Okay, and so the FW and the wild type you can also fit to dissociation. And that seems to be single exponential in both cases. Again, you have much more stably bound A3G for the wild type, forming these oligomers that stay bound even as you have flowed out all the ligand in it and you're letting it dissociate on its own. Okay, so we have to fit this to a biochemical model. This is the same model that I told you before for the polymer, in fact. So the model is simple. You have a biomolecular process where it binds to the DNA in some state, maybe a monomer, maybe a dimer, at some fast rate, K1C, that's proportional to concentration, and there's some K-1 rate to come off, and then there's there's another process that's not proportional to concentration, K2, that's the formation of oligomers, and some K-2. And that's going between the fast and the slow state. And again, you can solve this model if you make the approximation that there's a quasi-equilibrium in this state in between. And then you'll notice that I call these K-fast and K-slow, and that's not the same thing as K1 and K2. Okay? The reason is because the slow rate is actually fed by the bimolecular rate. So even though this rate doesn't depend on concentration, it's fed by a rate that does depend on concentration. And so you get a concentration dependence to K-slow that you can fit. And so you get K-slow is some K2 times this factor here. So at very high concentrations, this becomes concentration independent. But at low concentrations, it's limited by this K-fast rate, which is the normal bimolecular rate that you expect to see. So this is what you would predict if this is the model that we should observe in our data. And here's the data. So yes, the K-fast rate definitely depends linearly on concentration. And we can get our rates out of that, some K1 and some K-1, directly from this linear fit. And once we know K1 and K-1 from here, we can plug that into this equation and calculate K2 and K minus 2 for the oligomerization rate. And so we can do this for all of the mutants. And we actually, the K minus 2, we don't have enough data points down here at low concentration. We don't have enough singular noise to get K minus 2, but it's very, very slow. It's the hours or tens of hours time scale. Okay, so what are the results for the different mutants? So this is where it becomes interesting, and it was very surprising for us. So first of all, we see that the association rate for initially binding, the concentration-dependent rate, K1, does not depend on the mutant except for the FW mutant. So the FW is the monomer. So the one that's a monomer is faster than the ones that form dimers or other oligomers later on. The dissociation, this was what was really surprising. The dissociation is the same for all of them, even though they're wildly different mutants. But again, that's K minus 1, so that's dissociation from the initial state, not the oligomeric state. And then, of course, what we did expect is that the oligomerization rate, K2, is much lower for the FW mutant, for the monomer mutant. Okay, so we can analyze each of these separately. Here's the one that we thought was the most interesting. Why do they all have the same off rate? I told you at the beginning that it was a controversy in the field. Is this a monomer or a dimer when it binds to DNA? If it were a dimer when it binds to DNA, this would not work because we know FW is a monomer. So you can't have the monomer have the same rate as the dimers for dissociation. You've got twice as many binding sites. There's no way that's possible. So what this says is that it must be, all of these are a monomer in solution when they bind. So that resolves that controversy. We also see that the K2, the oligomerization rate, is much lower for FW, but it's not 0. So why is it not 0? Well, either the N-terminal domain mutant is not a perfect mutant, and it still can dimerize through the N-terminus at some point with some weak dimerization, or the C-terminal, or we've measured essentially the rate of C-terminal dimerization. We can't tell the difference between those in this experiment, but it's clearly much lower than the dimerization rate for the other mutants. And finally, why is K1 greater for FW? Well, we're measuring K1, and while we're measuring K1, we're actually observing oligomerization. So oligomerization is happening in real time, and so if it starts to oligomerize, K1 is going to be inhibited by the fact that there are other proteins already bound and blocking the binding sites, basically, in a way that would not be true for the monomers. That's our hypothesis anyway. We don't have any rigorous data to show that, but that's what we think is happening. So we said, okay, we see oligomerization. It looks like oligomerization is just dimers forming because the initial K1 is monomers. So the K2 must be the formation of dimers because that's the next order of oligomerization, and it's the same for all of them, of the mutants, except for FW. And so we said, so we asked our collaborator, why haven't you measured this in your experiments, right? Because they do experiments where they measure this enzymatic activity all the time. So why didn't they see this? So what experiment do? They do an experiment where they have a short DNA, and they have these CCC sites that can be deaminated, and they use uracil DNA glycosylase to chop it whenever it gets converted to a U. Okay, and so what you get is a gel that shows you the different lengths, and you can see whether one of these was chopped, or two of them were chopped, or none of them were chopped. And the problem is that as soon as you do the experiment and add this, by the time it would normally dimerize, it's already done this process. So that's why they never see it, because they're using such short DNA molecules, but they can't use long ones. So we said, well, why don't you try an experiment where you start with oligomers? So we said, why don't you first add unlabeled DNA, okay, so DNA that doesn't have the sites, and wait for a certain amount of time, of say 3 minutes, and then measure its ability to act on these substrates. And what she saw was for FW and wild type. So FW is the monomer, it can't form oligomers, and wild type forms oligomers. And so what you see is that FW is just as good as before when you have this unlabeled DNA incubated, but the wild type is almost non-existent when you have a high enough concentration. So it says that not only is this dimer very slow, the dimer is unable to perform its enzymatic activity. So instead of being a thing that scans along the DNA, it just sits there. Okay, so not only could it inhibit replication, but its enzymatic activity is gone. Our findings from this are, first of all, transiently bound A3G dissociates at a single rate, so we know that it must be a monomer. Mutating the C-terminal dimerization does not significantly slow the dimerization, so the C-terminus isn't playing as big of a role, but obviously it's needed for those big oligomers to form. And also that deamination by A3G decreases when it's pre-incubated with single-stranded DNA. So this means that wild-type A3G exists as a monomer in the absence of DNA and during deaminase activity. So when it's scanning the DNA, it's scanning as a monomer. That was a controversy in the field. Dimerization mediated by the N-terminal domain is sufficient to significantly slow the binding kinetics and to inhibit, and that dimerization inhibits the deaminase activity. So what does that mean about the moving along these tracks of DNA? So what happens in the virus? So again, in the virus there are 10 of these, but if you can form a roadblock with just 2 molecules, that's obviously much more significant than if you need 1,000 of them, because if you need 1,000 of them, it's not going to work. But if you only need 2, that can happen in the virus. In fact, 5 of them can occur in the virus. So in the virus, although there are only 10 of them, the concentration is very high because the virus is very small. And so in less than 1 second, they bind and they deaminate on the time scale of a minute or so. They can scan the entire sequence. And then after about 150 seconds, according to our measurements, that K2, remember, saturates at high concentration. So that's basically at the rate of K2. The dimers form, and those dimers can form a roadblock. Of course, you can get higher order oligomers as well that can form a roadblock, but all you need are dimers, is what we've shown. And that can act as a roadblock. So A3G monomers bind transiently and are highly mobile and deaminate. Then dimers form, and they don't slide along the DNA, and they act as a roadblock. And actually, I didn't talk about it, but correlation between replication studies. So if you make this FW mutant, or a very similar mutant to the FW mutant, so it can only form monomers, it no longer is able to inhibit HIV replication. So this dimerization that we've described, we know is actually functional in the virus. It's just that we didn't know what it was that was functional. That's what we've discovered here. With only 10 subunits, the ability of monomers to deaminate and dimers to form roadblocks is obviously important for A3G function. And so let me finalize by thanking my collaborators. So the initial work on the polymerases is with Professor Penny Boyning at Northeastern, and was done by Naba, and some of you have already seen probably his poster here, which describes those experiments in more detail. And you'll be able to talk to him again about it at the next poster session. And the work on A3G was done by, the AFM was done by graduate student Ran Hu, and Mike Morris is a postdoc in the lab, and done in collaboration with Linda Chellico at University of Saskatchewan in Canada. And thank you for your attention.
We have time for questions, please. Yes, please. Ask some questions? No? Yeah, okay.
In this plots, force versus extension. So what I'm asking is that by doing applying some torque and measuring torque versus rotation, do you think that some extra information can we get except this force extension plot?
So sorry, the question is about using force extension plots to understand DNA binding. So what's the question about?
So why always extension? Why don't we apply some torque and try to understand?
Right, why not apply torque? Okay, that's a good question. There are other people who have used torque. It's much easier in our experiments with optical tweezers. It's hard to rotate to find. You can make tweezers that allow you to rotate, but they're very complicated. So people use magnetic tweezers to do that. And there's several groups of people who study things like topoisomerases, which change the linking number. So it can be done, and other people are doing it. In this case, this process doesn't involve any kind of torsional change. So, in the cell, a lot of that torsional strain is relieved by enzymes that are responsible for doing that. Yeah.
Okay. Yes, please.
A3G is packaged inside of the virion. You said there are 10 copies inside of the virion.
Yeah, that's right.
And so it's bound to RNA while it's packaged inside of the virions. And at which stage of the reverse transcription is starting to be active? Just when DNA is formed?
Yeah, so it binds to the RNA. That's a very good point. So A3G is packaged in the virus, was the point. And the question was about the what stage does this deamination, does the enzymatic activity happen? And so, yeah, once it's packaged in the virus, it's going to be already bound to RNA. It binds strongly RNA, but it does not have enzymatic activity on RNA. It can't modify RNA bases. It only modifies DNA bases. So it's only after reverse transcription happens and creates single-stranded DNA that occurs, and that only occurs after it's entered the cell and become infectious, basically, because it has to have the NTPs to start that process. So binding, though, is initiated originally inside the virus, and it probably forms oligomers there that have to be somehow broken up, and we don't know how that happens.
So your model supports the fact that DNA synthesis occurs in an intact capsid.
Oh, does it support? Yeah, I see. So does it support the fact that the reverse transcription occurs inside the capsid? That's a good point. I completely agree with that model, that most of reverse transcription occurs inside the capsid. I think probably it's when it forms the double-stranded DNA that breaks apart the capsid, but before that, there's no obvious reason why it would come apart. And your point, I think, is that if it weren't inside the capsid, you wouldn't have that concentration of A3G, right? Exactly. It would just float away, so it wouldn't be there. So that's exactly right. That's evidence that supports the idea that it must already be, it must be occurring inside the capsid.
Thank you.
Okay. I have a question. Sorry. Can I ask you a question about the first part, about this Pol III? So my question is, you have essentially 3 proteins together. Let's only think 2 together, right? The question is the presence of another one accelerates the exo function or polymerase function. What if they will do separately? Will it work as efficiently?
Right. So the presence of the other one, all it does is increase the concentration of the other one at the site, right? But other than that, I don't think. Well, because they each have a DNA binding site.
Right.
So it can switch back and forth between those 2 sites. But what our study shows is that effectively each protein is acting like an independent protein. So the only reason it has to be there is, I think, to increase the local concentration of the other protein.
Okay. So, Prabal, please.
While along the ssDNA, the dimer or oligomer spool binding. So
yes.
How big is this tract, like the ssDNA? Long, and to move along the track of the ssDNA, there has to be some conformation where the monomer.
So, I'm sorry, so.
No, so you mentioned that in the monomeric stage, the A3G, it's probably moving along the ssDNA.
Right.
But when it
has to sample those states, but it doesn't move along them like, like a motor. It's definitely coming off and going back on, and and hopping, and that's actually, there are a bunch of papers, controversies about exactly what that mechanism is, but it's some combination of 3D and 1D diffusion. But the idea is that it must sample those sites. So it's a 9,000 base long sequence that it has to sample all the bases to find, within a few minutes, to find all those sites.
Okay. So let's thank all the speakers this morning.